The keynote speaker this morning, I'm really looking forward to this one, is Sabine Hauert, who is a Swiss-American who is at the University of Bristol in the UK, so she's obviously a citizen of the world. And she's been doing research into nanomedicine, or medicine using nanobots and swarms of nanobots, and I have no idea what that means, but I'm really looking forward to finding out. So with that, Sabine, please come up. Thank you.
All right, thanks so much, Mike, for the kind introduction, and thank you for the invitation to be here. It's wonderful to be at the Eclipse Con. Today I want to tell you a little bit about my work designing swarming nanomedicine. And really what it is it's a journey between swarms of flying robots to making things that are much, much smaller and work in very large numbers. I'm a lecturer over at the University of Bristol and at the Bristol Robotics Lab. And when I got the invitation, I was quite excited because actually the way I started my research career, so this is just a little abstract from Uppsala in 2004, was doing research for someone who was doing aspect-oriented Java, and it was a plugin for Eclipse. And if you go into the acknowledgement, there's little old me somewhere at the bottom. So I'd like to think that maybe Eclipse is what got me on this research path. So maybe keep that in mind. It would be nice to circle back and put this all together. Oh, so maybe, did you remember that? Well, I'll tell you the story about that. What I've been focusing on now is really this idea of swarming. So if you look at flocks of birds, they can do these beautiful, complex behaviors. And yet each individual bird is not following a complex set of rules. These flocks have features that I think are really interesting for real-world applications. For example, you can keep adding birds to the flock, and the flock is going to continue to fly, so it's scalable to large numbers. If a bird falls from the sky, the whole flock doesn't crash to the ground, so it's robust. And together they can do more than the individual parts. So for example, they're better at avoiding predators. And we want to take inspiration from this to bring it into the real world in terms of engineering. And the challenge with that is that we've really got a puzzle, because very often you know what the group does. So for example, the birds are able to flock. That's a group behavior. But if you take a bird from the sky and look at it, there's no way you're going to understand how that flocking happens. And so the challenge is, given a group behavior or a swarm behavior that you want, how would you go and design the individual agents? And we really need to set up a set of tools that would allow us to do that. The first application I was interested in studying was over at EPFL in Dario Floriano's lab. And it was to create a swarm of flying robots to create communication networks in disaster scenarios. So the idea was that you would have these rescuers arrive, they'd throw these flying robots in the air that should be able to distribute over the area on their own, do something a little bit swarmy, and then they should be able to communicate through that network to other rescuers or to victims in that environment. So that was the general idea. And we were using flying robots that were about this big. They were made out of foam, quite safe. They only had one propeller at the back, and they had an autopilot and a Linux computer with a Wi-Fi dongle that allowed us to control the robots. The challenge was, again, you know the group behavior. You know you want to create communication networks using a swarm of robots. That's your group behavior. But how should you program the robots so that when you throw them in the air, they have that emergent property? And so the way we started to do this was to figure out if we could use machine learning to come up with these programs for the robots automatically. And the way we were doing that is we had a little program in our robots, which was a neural network, an artificial neural network. And all it took were inputs that were the compass of the flying robot and communication with neighboring robots. This wasn't even relying on GPS, so it was really using communication as a rubber band to have these robots coordinate. And then, based on those inputs, you basically get a number of multiplications and comparisons between those inputs. And then at the output, you get a turn rate. So how much should that flying robot turn? And we're only acting on the turn rate because if you modify the speed too much, your planes drop. So we're just playing around with the turn rate. And then what artificial evolution does is that it essentially takes all those links and automatically comes up with what those links should look like, what the weights should be. And this is what we discovered in simulation. So here you've got a rescuer, another one. They have a limited communication range, which is this blue circle. And if you throw them in the air, this is simulation again, I've got 20 bots. They create these chains, and then these chains synchronize, and they go from left to right so they can search for the second rescuer. And once they've connected, they can maintain that connection in a certain way over time. And so we had this idea from evolution, but then we needed to go and push these things to reality. So here you have 10 of our flying robots, and I'm behind the camera here. This is Sabine. And so you could really tip the nose to the air. These robots would take off. And then we could start to implement some of the ideas that we discovered using this artificial evolution. So here what you're going to see are 10 of them in the air. They're about bird size, so I challenge you to find the birds. There's a couple, usually at the top there, quickly zip by. Sometimes they follow the flying robots, so we've been wondering about what kind of thing we could do to have nice interactions between the birds and the robots. These are GPS trajectories between the two rescuers. So that's the chain that you saw in that simulation before. And this is them synchronizing. So they're all moving with the same heading at the same time. And this is something that we implemented based on what evolution had come up with. And it was fun because what evolution came up with were things that we hadn't thought of when we were trying to program these by hand. So it's a pretty interesting creative process. The other thing that you can do, beyond automatically designing these swarm rules, is to take inspiration from nature. So for example, what you have here are pheromone paths laid by ants. So you know ants are very good at creating a trail to your picnic table. Well, it turns out that if you use rules that are ant-inspired, you can do something similar with these flying robots. This is in simulation again. You've got your two rescuers. And just like those trails, they're creating paths that reach out in different directions. The green ones are ant robots. The red ones are the environment on which they're depositing these chemicals or pheromones. It's not real chemicals. It's all virtual. And then they can create that path, maintain it, and then once the communication is done, they can all come back and land at the rescue site. The other source of inspiration was the flocking. So you saw the flocking behavior before, and there's rules to achieve flocking-like behaviors that's typically used in the video game world, actually. People really like to see flocks or herds or crowds of people. And the way those work, let's just focus on one robot here that has a limited communication range. So it's only looking at a couple neighboring robots. And what it's going to do to maintain a flock, to stay together, is it's going to be attracted to its neighbors. And that makes sense because you want to make sure that flock stays a cohesive entity. It's going to be repulsed by those robots as well, because if everyone is attracted, everyone collides, and that's not very helpful either. It's going to try to align its heading, so they're all facing more or less in the same direction. And then what we did is we gave it a migration point, that star there, that says essentially we want your flock to go to that star. So that allows us now to have a controllable flock that we can direct. And so what you're seeing here are the GPS trajectories of the real flying robots in the air. And you can see it's very messy in terms of their topology. But over time, we get a circle. You can see the wind, by the way. You see how he moves at different speeds, depending on what direction it's facing. So at some point, you get a circle. And that's what we had predicted using our models. So very often what we do with this swarm world is we design the rules, we test them in simulation. That's why you saw all those simulations before, to see if we could predict what the group would do, and then we could throw them in the air. This is our interface on the computer, so we can see who's talking to who. We have a lot of information about their position as they go. And then at the end of the experiment, we can bring them back. So that was fun, but really, those were 10 flying robots. And I always thought that swarms needed to work in the hundreds and the thousands and the millions. And so I got very interested in the idea of looking for a new platform with which we could play with this idea of swarming. So here what you have is essentially the start into the field of nanomedicine. So nanoparticles, because of their size, which are nanometer-sized, are very good at leaking out of vessels into tumor environments. And so that makes them interesting delivery devices for treatments or for imaging so that you can see the tumor from the outside. And these nanoparticles, so they leak out of the vessels, they arrive in the tumor environment, which is typically diffusive. That means the particles move around randomly in there. Those particles work in the 10 to the power 13. And so there was the one with the many zeros that I was looking for. And even beyond that, these particles have a lot of interesting features. So you can change their size, you can change their shape, you can change their charge. That's going to change what they stick to. You can change their material. Some of them are energy receptive, so you can use magnetic fields, or you can use light to activate these particles. You can coat the particles with little molecules that allow these particles to bind to cancer cells, for example, or to bind to each other, and then you get self-assembly, and you get disassembly. And you can load them with drugs that you can release in a more or less controlled fashion. And so there's a lot of knobs that you can turn on the design of these nanoparticles, a lot of different ways in which you can design them. And the question then is, well, how would you possibly make nanoparticles swarm? Well, I guess with the birds, the birds probably had a brain that was telling them how to react to the neighboring birds. And the flying robots, well, I programmed them to do a specific behavior at a specific time. So it was their program. With the nanoparticles, well, they don't have a brain, and we can't really program them. But what we can do is change the design of the body of these particles. So we can turn the knob on the design of these particles so that when we inject them and they arrive in the tumor tissue, they do what we want them to do as a collective, as a group. And the reason that's interesting is because of all these keywords that come from the swarm world. So if you look at bees, they're very good at making decisions about the next location of their nest. What if you could make nanoparticles that could make decisions about the state of a tumor? Ants, before you saw those trails, they're actually very good at optimizing things. So those trails very often are optimized to go where the food sources are. What if we could make nanoparticles that could optimize trails or increase imaging to areas that we're interested in the tumor? And what if you could amplify the delivery of a drug using things that are swarm-based? There's so many examples in the swarm world where you could take these keywords and try to put them in a context of nanomedicine, and those would become interesting treatment applications. And it's not a completely crazy idea. So what you're seeing here is work done by Sangeeta Bhattia's lab over at MIT, where I spent 3 years. And it was really a wet lab that tested these systems in living systems to see if they worked, if they could shrink tumors. And so here you have a 2 nanoparticle system. What they do first is they inject these blue ones. They arrive through the bloodstream, leak out from the bloodstream into the tumor tissue. And those blue particles are made out of gold. So if you shine near-infrared light, a laser basically, on the tissue, those gold particles vibrate, heat up, cause damage to the tumor tissue. And then they send in these pink particles, which are liposomes. They're fatty bubbles, essentially. And those are the ones loaded with the drug. And those liposomes arrive through the bloodstream, leak out, and then they're engineered to bind to the damage that was just created in the tumor tissue. And you get 40-fold increase in the amount of drug you can deliver in that way compared to those that don't somehow communicate through the environment. So just like ants deposit chemicals so that the other ants can sense the environment, these blue ones come in, do something to the environment, and that's going to change the other particles' behave. So we're starting to see these systems tested in real in vivo settings, and you do see an increase in the amount of drug that arrives there. There's also lots of other examples where particles self-assemble and disassemble, and that changes the way the particles move and improves treatment and imaging applications as well. So that's the big picture of what we want to do in terms of swarming nanomedicine. But before we get there, we really need to, show that we understand the system. And so what we did is we spent quite a bit of time just looking at one scenario so that we could start modeling these nanoparticles in tumors and try to be helpful to bioengineers now. So this is our test case. We've got a vessel. We've got cancer cells. Those cancer cells on their surface are overexpressing receptors. That's what I drew as a red line. And our particles have a similar coating so that they can bind to the cells. And when they bind, they get eaten up. So the group behavior here is make sure that you're killing all the cancer cells. That's a group behavior because you can't have a single particle possibly achieve that. And we only have two parameters, or two knobs that we turn on the design of these nanoparticles. The first one is the size of the nanoparticle. That changes how fast it moves. A small particle is going to zip through, whereas a big particle is going to move very slowly. And how sticky this particle is to the cancer cells. And because we really wanted to be helpful, we have numbers from the literature, so we know things like how much you inject, how much arrives in the tumor, it's a certain percentage. Then we can zoom into that tumor, and we can take a slice out of it. And then in that slice, we can't possibly model the whole tumor, because even though you have that 10 to the power 13, modeling that 10 to the power 13 is quite tricky. So we look for what we call a worst case scenario. So what's the hardest thing to do in that tumor if your goal is to go deep into that tissue? And it turns out to be we're looking at the furthest point from any vessel in that tumor. So it would look something like this. You have a vessel, and then you have 20 cells in a line. That's around 200 microns to give you a scale of that. And our goal is to make sure we kill those 20 cells. And in each one of those boxes, we've basically got a well-mixed environment. We use stochastic simulators. And what happens is the particles can either bind to the cell, unbind from the cell, and when it's bound, it can get eaten up by the cell, and once it's eaten up, it delivers its drug. And we can count how much drug is delivered, and then that tells us if we've reached a lethal dose. And then the particles can move randomly from box to box. That's the diffusion aspect. All right, so we're trying to kill these 20 cells, and this is what we get as a result. So if our goal is to kill 20 cells, that's dark red. And again, we have two parameters: how fast your particle is, and how sticky your particle is. And these are all numbers from the literature. And it's interesting to see that it's a hard task. First of all, most of these particles don't kill 20 cells. And then you can actually go and look at the individual boxes. And if I just pull them up here, first of all, there's lots of different group behaviors. And just look at A at the upper right there. That's a particle that was very sticky. And because it was sticky, it leaked out. And the first cell it encountered ate it up. And so that means that all the particles are essentially getting eaten up by the first cell, and they never go deep into that tumor tissue. And that was really counterintuitive, because if you're a bioengineer, you want your particle to be a cell killer. So you want it to go and get eaten up by a cell. But if you do it too well, in terms of group behavior, swarming, that's actually a poor choice. We can then use these simulations to come up with better ideas. So what if we let our particles leak out, but we have a shell on them, and that shell essentially would prevent them from sticking, and so that would allow them to go through the tissue, and then you would take that shell off. And you can do that with light, you could do that with pH. We draw enzymes as little Pac-Mans because they go and cut stuff up. And then if you do that, then they would become sticky again and be able to be eaten up by the cells. And you can use, again, these simulations to come up with how long you should wait. So we thought you should wait 17 hours. 17 hours before you take that shell off. And it turns out that if you do that, for all the sizes that you saw, and for all the stickinesses, you kill the 20 cells. And not only that, but you can change the scenario in a variety of ways, and you still kill the 20 cells. So these are things that are very concrete for bioengineers, and that they can use today in terms of tricks that might get them deeper tissue penetration. And it turns out that people can make these particles with these special shells. So this is work by Paula Hammond's lab, and she has these particles which have this shell. There are these blue squigglies. And then when they arrive in the tumor environment, because of the pH in that tumor environment, they're going to lose that shell, and then they become positively charged. And positively charged particles like to get eaten up by cells. And it turns out that in her paper, although that wasn't the intention, she gets her particles going deeper in the tumor tissue. So that was really us spending 2 years learning the basics of nanomedicine and trying to make a model that we thought would be helpful for the bioengineering community. But really our objective was to do these swarming nanosystems. And so we needed to be able to explore, turning all these knobs in lots of different ways. And there's many of these knobs. So it turns out that the computational space is quite tricky. And so what we did is we designed a game called NanoDoc that allows us to crowdsource this exploration step. And so you can go and play the game on nanodoc.org. I'd love to help with this. So if anyone is passionate about developing potentially games for cancer, let me know, because it's something that we definitely want to look into. And so this is what the game looks like. It's really one part are the scenarios that we want to solve, the things that the bioengineers would submit. And then on the left, you have your editor, which is essentially where people go and design different types of nanoparticles. And when you click inject down there, that goes back to our scientific simulator that uses the numbers that were relevant to the community. All right. So the things that we can do with this is that we can look at cancer cells. So this is how we define a cancer cell with a lethal dose. That's essentially how much you need to kill it. We need healthy cells. Do you know why we need healthy cells in this game? The tricky thing is that if you don't have healthy cells in your scenarios, the solution to every treatment is just to dump loads of drug on it. And so you want your systems to be specific so that you're not causing harm to the healthy tumor tissue, or the healthy tissue. You can change the treatment dosage. You can change when you inject these treatments. You can combine the treatments. So remember those pink and the blue particles. I really want to explore those types of synergies. You can change the size of the nanoparticle. That's going to change how fast it moves. You can have these cancer cells with a border now. And because they have a border that's letter-coded, you can have those nanoparticles that have a similar coding. So it's also letter-coded and color coding. You have a certain strength in terms of how sticky they are. You can load those particles with a cargo, change as well how that cargo moves and what strength it binds with. And then you can make smart materials, where essentially if something binds to that particle, it's going to release something in its environment. And so if you're a roboticist, you get excited by this, because all of a sudden you have something that can sense its environment and put a signal out in its environment. And the thing that was missing is that we couldn't really change how these particles moved. They were just moving randomly. They were diffusing, whereas my robots, the flying robots, before I could tell them to go left or right. The ant-based algorithms, the ants go left or right, depending on what's on the ground. And so how could you possibly control these nanoparticles so that you can tweak their motion? And it turns out that you can do that with self-assembly. So if you have particles that come together or basically break apart, you're changing their diffusion dynamically. So it's not so much about telling them to go left or right. It's more about telling them to stop and to go. So we spent a lot of time disseminating this. So I run robohub.org, which is a nonprofit to connect the robotics world to the public. And we put that out there, and then we did a couple things to also get the community excited. So for example, you get little diplomas when you learn about the skills. So all those things that I showed you in the previous slide, those are skills or levels of the game, because you can't possibly learn all of this in one go. So basically, you get little diplomas at each stage, and you can go ahead and tweet those and say that you're a nanodoc or you're proficient in certain of the skills in that world. Obviously, nanomedicine is actually quite hard, and cancer treatment is a very serious issue, so we have educational material as well to go with that. So we've had around 100,000 simulations from the crowd, and actually now we have a huge amount of data, and we're just combing through that to see what it is that the crowd's doing. Something that's quite fun, actually, is my whole PhD was about automatic optimization, right? The computer came up with the solutions. Now here we have a system where the crowd is coming up with the solution. And I think where we're going to is figuring out how you put both those things together, because the crowd in a couple clicks can optimize something that would typically take us a long time to go over the whole parameter set. And so we're going through this data, trying to understand what the crowd does so that we can make better computer optimizers, and also understand what the benefits of one versus the other are. I love both methods, so no preference yet. So this is a solution, a set of solutions that were found by the crowd. And here what you have is essentially a rare event, that blue cell, and then our goal was to make them accumulate particles around that blue cell. And that could be seen as a way to amplify the signal of that blue cell. It could be a stem cell, it could be a mutation. It's just something we want to detect in that tumor environment. And there's two interesting things here. The first is that particles leave that vessel at the upper left, but it really looks like they're propagating from the other corner, which is counterintuitive in terms of the dynamics. The other thing is that all these solutions needed two particles. One particle binds to the blue cell, releases a signal, and that binds to the second particle. that releases a signal and accumulates in the surrounding cells. And so that's essentially a system where one particle communicates to another particle. And that's something we had never taught the crowd to do, and something we were excited about, because it does show that they can do out-of-the-box thinking. And not only that, you can just throw them new challenges. So actually, the new challenges don't even have the same fitness functions. It's not about killing the cancer cells anymore. It's about accumulating in certain areas. And you don't need an optimizer behind that to figure out what the right algorithm is. The crowd just picks it up and runs with it. What we do now as well is that we use these simulations, basically play our own game to come up with more swarm-like behaviors. So this is an experiment where we went back to these ant algorithms and said, okay, well, what if we basically simplify these algorithms down, and we simplified and simplified. And so here what you have is essentially, again, that rare cell in the bottom right, and the vessel, which is the input to the nanoparticles. And you can see they're creating these trails between the 2 points. And it's a very basic mechanism of basically sticking to a cell, releasing something in your environment, and then the other particles need to stick to that cell. So you get some reinforcement in there. So everything you saw in the nanoparticle world was simulation up until now. And really, just like with the flying robots, we want to be able to do something in reality so that we're actually having an impact. And the first thing that we did was to actually try this on robots. And so here what you have is a kilobot, because Mike Rubinstein and Radhika Nagpal, who made this over at Harvard, made 1,024. So instead of a kilobit, you have a kilobot. They're coin-sized. You can learn all about them on kilobotics.com. They're actually open hardware, which is really exciting because that means that we get to make them. And I used to go to their lab every week, so they've also been extremely helpful in what's going to follow. And these little robots, they needed to be quite cheap to be able to get 1,024 of them. And if you're going to control 1,024 robots, that means you never can touch them to turn them on, to turn them off, to reprogram them, because that just wouldn't scale, and you wouldn't be able to do experiments with 1,000 robots. So the really nice thing with these robots is everything is done using infrared light remotely. So I basically just have a dongle in my computer. I tell them to start, to stop. I reprogram them using light. They have 2 vibrating motors. That's what you see on the side. They look a little bit like batteries. That allows us to control their motion, but since it's vibrating, it's quite noisy, both in terms of sound if you have 1,000, and in terms of how they move. They have an LED, and that allows us to see what state they're in, what they're thinking. They have an ambient light sensor, so you can project light patterns in their environment, and they can interact with that. And then they have communication, infrared as well, within 10 centimeters, so they can talk to robots in their neighborhood. And if you're wondering why there's a hook, well, again, you can't charge 1,000 robots if you have to put them one by one. So we just push them to a metal sheet, put another metal sheet on top, and then you can parallel charge many of them. So we were really excited by these robots, because the simulated world is you do make assumptions, you do make simplifications, and you could make the simulations better and better and better. But really, with these robots, with 10 lines of code in just 10 minutes, we can test these algorithms on a physical system. And sometimes the assumptions you had, you can see them break down in front of your eyes just looking at these physical robots move around. They're a nifty way to think about dynamics. It's very visual. So here, what you see are. This is just the first experiment we did over with Radhika's lab. And so these robots that are all alone are going to be our cancer cell robots, and they can communicate within that range. So you bind to these cancer cell robots through communication. And then you've got your nanoparticle robots on the left. So here you can see they're kind of moving around. Then they stick to the cancer cells. This is a case where the particles was very sticky. So if you remember before, sticky particles were bad because they never go deep into that tumor tissue. And very often we actually run these in the dark because they look like fluorescent markers, which is what the bioengineers would be seeing under the microscope. So they turn purple when they've been eaten up. And it's kind of fun because there's lots of room between those 2 cells, lots of room for the particles to go through, but they never do because of the nature of diffusion. And you don't really think in terms of diffusion. You think, why don't they just go through there? But they don't. That's from an actual microscope. Then we had that strategy where we let them go deep into the tumor tissue. Actually, what we're doing here is they're not as sticky. So this is a less sticky particle. And if you look at the upper right, there's a cell all alone, and then particles zip by, and they never stick to it. And that kind of makes sense, because we've tuned the binding down so much, it's become hard for them to stick to the cells when they encounter them. Eventually you do get one, and that's really just because it's stuck next to it for so long. And then we tried the third approach, which was diffuse, and then take that coating off and let them stick. So here they're diffusing, and then you just turn them on, and then they need to start sticking to those cancer cells. And again, look at the one at the upper right. So here they're just diffusing, no one's stopping yet. And then at some point we turn the bonding on, and you can see they'll all stop around that deeper seated cancer cell. And we thought, we knew this from the model, so we didn't learn anything in that respect, but we thought maybe we can inject way less particles with this strategy because it works much better with the deep-seated tumor cells. And it turns out that you can go back to the numbers that are relevant for that field and show that you don't need to inject that much. So just seeing that happen does give you interesting ideas. What we've done since then is I've been at Bristol for 1.5 years, and we built 1,000 robots thanks to these open hardware designs. So this is our swarm of kilobots. And they were all built by students, so it was essentially a sweatshop for 2 weeks, but actually that was the funnest week of my year. And the thing we've really been studying, you can see our arena has this projector, so we can project these light patterns. And what we finally figured out is how to scale the nano world to the robot world, so that if you take a nanoparticle and blow that up to a centimeter-sized robot, there's a scaling factor there, and if you do the same thing with time, there's a scaling factor there. And so what you're seeing there is a scenario that is proportionally similar to what you would see at the nanoscale within cancer cells. So we're just now starting to do these experiments to visualize these dynamics. And then the final step here is really to push these things to reality. So we do a lot of work trying to visualize these nanoparticles under the microscope. So here, this is a device that was made over in Sagita Bhatia's lab. And so there we had one channel. It's really just a micro-scaled channel in plastic, essentially. The red are nanoparticles, so they're full with nanoparticles. You can think of this as our vessel. And then the green channels, each green dot is a cancer cell, and they're in gel, so the tissue is a little bit tumor-like. And we can see where these nanoparticles go when they leak out of the vessels. This was actually tricky for us to maintain because you had living cells in there. And so as engineers, what we started to look into is how can we make devices that are really quick to prototype and where we can very, very well control everything in that tumor-like environment without them being living cells, just so we can visualize these dynamics in a more repeatable and easy way. So what we do now is we 3D print scaffolds, and then we pour our PDMS, so that plastic-looking element there. And then you can make your microfluidic devices. These are what our particles look like under the microscope, so it's quite easy to see the individual particles. So this is something that we get from our channels, where now instead of cells, I've inverted the colors, by the way, so green is particles now, and red are microspheres that are cell-sized. So they're essentially fake cells in our channels that we can functionalize and put things on their surface so that we can start to understand where it is that these particles are going. And so just in terms of nanomedicine, I want to wrap this part out, and then I want to branch back into the robotics world. What we're really trying to set up is this idea of systems nanotechnology or swarm engineering, where you think really first about what your group behavior is. Is it to kill all the cancer cells? Is it for them to create trails? Is it for them to do decision making? And then from that group behavior, you need to figure out the rules for the design of the individual particles. And we use lots of simulations and computational tools to visualize that and to be a little bit predictive. But then we try to push this to reality, and we do that using robots to be able to quickly visualize dynamics on physical systems. And then we're doing that in the nano world under the microscope with some of our microfluidics. We also work a lot with researchers who are more translational, who can actually do these experiments in vivo, so that if we have something helpful, it has a potential to go to the clinic. And the thing that I'm excited about is this pipeline is the same pipeline that you need for the robotics world. And as we discover how you control thousands of trillions and millions of trillions of nanoparticles, I think we can take those lessons and bring them back to the robotics world and stop assuming that you're going to have very precise robots that are going to do the tasks in a very predictable way. Now all of a sudden we can start thinking of robots that do work in the thousands and can still achieve meaningful tasks. And this is really a different way of thinking. What you're seeing here are 30 of our robots. And we've programmed them to go straight. So this is essentially our robots trying to go straight. So if you were to take the average, they would be going straight, and you have to stop thinking of them as individuals and really start thinking of them as collectives or swarms. And so how do you control these robots? And really, I'm convinced you won't be able to calibrate thousands and thousands of robots if you're putting them outdoors. So figuring out simpler robots that still work to do a task, I think, is the way to go with these very large systems. And I just wanted to show you results from last week where we've taken the AMP-based rules that we're creating those trails in the nanoparticle simulator, and we're putting it on these robots here. So these are the real robots in our arena. And you've got your rare event. That's the thing that you want to create a trail to. And Perra is essentially the vessel. So you can see his hand go in because he's putting these particles in, these robots in one after the other. This is random in terms of its motion, which is the easiest thing for us to do. And there's lots of, it looks very organic, doesn't it? You can see them kind of twirl around each other. They're doing some really interesting stuff that I have no idea how you would model that using a computer program. And so then you've got your rare event there. And at some point, you're going to start to see this trail form. Diffusion is slow, by the way. All right, so there's one of them that's connected. And it's interesting because as soon as you start getting your trail, the trail happens very quickly. And so this is really just a couple lines of code to get this up and running. And I'd be really interested in seeing how you could use these type of techniques to have robots that can navigate a forest and find an element of interest, to have robots that go up and clean an oil spill. So I think we're really starting to think, if you have 1,000 really simple robots, what is it that you could do in a real world setting? So with that, I want to thank a lot of people. So my students and postdoc and collaborators at the University of Bristol and the Bristol Robotics Lab. A lot of the flying robot work was done with Dario Floriano's lab over at EPFL. The nanoparticle work, a lot of it was done with Sangeeta Bhattacharya's lab over at MIT. The first kilobot work was done with Radhika Nagpal at the VIS. We use a stochastic simulator from MIT called SSC for the Stochastic Simulation Compiler, and funding from the Elizabeth Blackwell Institute and the University of Bristol. And hopefully we'll be able to thank the players. So I look forward to processing all this data and seeing what discoveries they've made. All right, and so with that, I've left a bit of time for questions. So it's open to you. So he's asking if there's any challenge in cleaning up after the robots. Do you mean the robots in the tumor tissue or the bigger robots on the arena? The nanoparticles, right. So the general idea is these nanoparticles need to go there, deliver a drug, and then that's the goal, is to kill the nanoparticles, kill the cancer cells. A lot of work does go into actually figuring out the toxicology of these particles to make sure they're going to the right place and that they have no bad side effects. And that has to do with the cleaning up aspect, is where do they go afterwards. And these nanoparticles need to go through the same licensing system or FDA approval as any other drug. And with that respect, these ones, for example, go more to the tumor than the healthy tissues. That's one of the benefits, but it's different for every particle. So for every particle, you actually need to go through that process, but that's a really important question.
Take the microphone over there.
Sorry. So the robots move on a plane which is two-dimensional, and also your simulations looked two-dimensional, but the tissue is a three-dimensional space. So can you project it easily, or is there?
Yeah.
Yeah.
That's a very good question. So our regions in the simulation are actually three-dimensional. So we do model three-dimensional diffusion within those well-mixed regions. The robots are definitely 2D. We'd love to make a 3D version, actually, where they're in a tank, and so you have some 3D motion there. Going from 2D to 3D is really important, and obviously things change when you are in the third dimension. So you do need to consider that third dimension carefully, and I think that's part of why we're going to these microfluidic setups so quickly, is because I think seeing the fact that the actual physical system in action is going to be the best validator, rather than over-engineering the simulations before going to that level. But we take into account the 3D in the simulation, but not on the robots. Mm-hmm.
Already turned on? Yeah, okay. So the overall approach that you take implies that delivering the drug actually solves the problem of fighting cancer. Is that right?
Yes, but different cancers will have different drugs, and for some of these cancers, you don't necessarily know what the right drug is.
Okay.
So, yeah, I'm assuming that you know what your cancer cell population looks like, and you know what drug would do that, and I'm trying to figure out what the delivery device should be. But then there's a lot of work that needs to go from the biologists and the clinicians to have the finer granularity information for that. Yeah, that's a very good point. Yeah, I have simplified to a large extent what cancer is, because there's so many different versions. And so this is definitely not a one-size-fits-all. We work a lot with brain cancer surgeons right now, which is completely different because you don't have this idea of diffusion, and the particles don't arrive through the bloodstream to the brain. And so there you're dealing with, basically you're injecting the particles directly, and you have completely different dynamics, and you're looking at centimeters instead of microns. And so, yeah, each one of these is different. But having these models is actually quite helpful because they don't necessarily think in terms of the dynamics. They tend to engineer the particles themselves. Mm-hmm. Sorry, I hope I didn't miss anyone.
Sorry, I have another question. So if, suppose you get a couple of millions of funding and you could spend it among the people sitting here, so on the software developers working on Eclipse and related software issues. So what would you spend it on?
So the two parts. One is the simulations. So making better simulators, because right now we've really just scratched the surface in terms of what we can model, but there's so many parameters. So right now we're very high level, right? The size, if you're sticky, how sticky you are. The parameters make sense for the clinical world, but they don't tell you what that molecule is, or they don't tell you what that specific material is. And so there's a huge set of simulations that we could do from this very high level to the lower level, so that at the end you would pop out a particle that you could potentially test. So I think that aspect is really interesting. And the other is NanoDoc is in need for love and care. So I think really building up a tool that would allow the bioengineering community to play with these models in an easy way, have the crowd that can put in their input, and have that's really nicely usable is something that I'm really keen on pushing. Yeah, in general, there's a big need for in silico, so computer simulations of these medical systems.
So what's the biggest swarm size you've been dealing with the nanomedicine, and how does it scale, or what affects its scaling?
Yeah, that's really interesting. So with the actual nanoparticles that you're seeing under the microscope, there we're dealing with, the 10 to the power 13 numbers. So they're huge, huge numbers. With the robots, we've built 1,000, so we can work with 1,000 easily in a laboratory setting. We don't really know how to push that outdoors. But it's fun because it's completely changing the way we were thinking about scalability. Because it used to be that we would prove our algorithms on 5 or 10 robots, like you saw with the flying robots, and say, oh, it works, and it must scale to thousands. But the reality is, when you have 1,000, so many are going to fail, and they're completely different noises, and it's just so different that our algorithms don't necessarily hold for those large numbers of robots. So the team at Harvard, they have a really nice demonstration. It's a science paper where they create shapes. So essentially you've got a blob of robots. You put a robot in there and it's the seed of a shape, and then the shape starts growing. And what they discovered is that faulty robots somehow impede the development of that shape. And they had to put an immune system somehow in their robotic collective so that you can figure out who's behaving, who's misbehaving, and be able to handle those large numbers of robots. So it's a wide open research question, essentially, is how do you scale to huge numbers of robots in reality? But the nano world does that just because they're meant, it's chemicals, they're cells, they're meant meant to work in those large numbers. Yeah. Does that answer your question?
Yeah.
Okay.
And I just wanted to say, I'm really jealous. I always wanted my own army of robots.
They don't obey me very well. Actually, it's funny because when I do demos, I'll have a scarf very often, and they have a hook. So I've had demos where I'm, playing around with the robots, and I get up, and they're all hanging around my neck, which is a very mommy-like behavior. But yeah. Mm-hmm.